G'day guys, this is a quick one in between my two live streams. Let me just bring it up on screen. Okay, so this is a study that actually is looking at the important differences between human and mouse APOE gene in relation to Alzheimer's disease. For a long time, I mean, most people that argue against a high-fat diet, and the issues about Alzheimer's and all these things. I mean, we know that the chow is full of seed oils and sugar and all that, but they keep on promoting the idea that pretty much, we can use rats, the mammals, and we can basically, extrapolate from that if it affects them in a certain way, it probably affects us and all that. Anyway, let's just look at what just the abstraction. This is just a quick little video. Okay, so it's telling us APOE encoded by the APOE gene may plays an important role in the pathogenesis of Alzheimer's disease. I disagree with that comment because it's ridiculous. Never been proven. There are more APOE 3s than 4s that basically have Alzheimer's or dementia. The only difference is you're more susceptible. Yeah, if you've got the ancestral APOE 4, and the only reason you're susceptible is because of the clearance of beta amyloid is slower, but that can be rectified with more animal-based diet, which is taurine, which clears out beta amyloid. And if you're actually having a species-appropriate diet, you will get more taurine. So, 1 point for the carnies. Now, they go, variant is strongly associated with Alzheimer's disease. Yes, associated, but not proven to be the main driver. No, it's the diet or the crap that people eat. Anyway, APOE promoter polymorphism has been reported to associate with high AD risk. Mouse models of APOE expression have long been used to study the pathogenesis of AD. We know that. They like using rats and rodents. Nothing like a rat study. If your name's Paul, you love them. Yes. Elucidating the role of APOE gene in AD requires an understanding of how its regulation differs between mouse and the human APOE genes. You would think that they would have actually focused on that right at the early stages of them doing research on this gene and trying to extrapolate from these little poor rodents. Okay, how the difference influences AD risk. We compared the structure and function of both human APOE gene promoter and the mouse gene promoter. Okay, so slightly different. I can't remember all the words that are part of it. Okay, now what did they find? The homology, that means the similarities. It's a Greek word, homologia. Omeo comes from the word omeo, which is similar. Is less than 40%. Yeah, it's only 40%. Oh, shit. There's a 60% difference. Oh, that really would fuck up the studies, wouldn't it? I couldn't. No way. These people know what they're doing. Remember, some people are rats. And because they're rats, they can identify with these studies. Anyway, we'll ignore this more upstream species transcription. Yeah, just genetics, transcripting, blah, blah, proteins. Anyway, we don't kind of give a shit what proteins they transcript. Functional analysis revealed both similarities and important differences between the two sequences. Fuck yeah. Significantly affected by human versus rodent cell line origin. We likewise probe nuclear extracts from the several cell lines of different origin, erythrocytes, glia, and neural, the mouse brain with specific blah-blah-blah fragments. Each fragment share DNA protein interactions with the other, but notably also bear distinct factors demonstrating by gel shift in south-west analysis. So, it's, those gel phoresis that they actually, for the genes, and then they look at and compare the two, those strips. That's what they're talking about. Shit. And from here on, we determine possible identities for these distinct factors. These results suggest the regulation of mouse and human APOE genes may be significantly unique to justify the use of both the human APOE promoter sequence in transgenic rodent models and non-rodent AD models for studying factors involving the AD pathogenesis. So basically what they're saying is that, there's so much difference between the two species. There's only 40% similarity, 60% difference, and a lot of different proteins are actually transcripted, are completely different. So the way certain nutrients and things like that operate, or the way things function and express within the brain, tissue, and all that, is so different that they're actually proposing that the only way we continue in the future studying is really in a non-mouse model, looking at, at that, which is human or another animal that is a transgenic, or to basically, we need to focus on doing real research with transgenic rodent models. There's a problem with that as well. The obvious model thing is that this is a reductionist nonsense, because, okay, you create a transgenic mouse that actually has the human form, so the structure, the gene promoter, this one, which is the human form, and not this one, which is the mouse form. Let's say you change that. Is APOE the only thing that's interacting? We know, I've explained it before in my other APOE thing that I did with PIM. We've got, for instance, from the HDL, the type of that will interact with other lipids. So obviously, if you change the thing in the mice, you still haven't changed everything else. You're still going to be interacting between things that are very different in general. So you've done one component and you're trying to extrapolate in a reductionist way from one component. That's just nonsense, but this is sort of what they're promoting. But what I'm trying to show you guys here is that you cannot extrapolate from those fundamental differences. So even if you create a transgenic mouse that has the APOE, expresses APOE in the same way as the human, our brains are different, our whole other parts of our biology are so different, this gene will interact very differently with the other genes within the mouse compared to a human. So basically it's just another bloody way to basically try and actually say, okay, we're fucked up until now. We've basically been doing dodgy studies that don't really translate to humans. This is why all those medicines that came out of these studies failed, clearing beta amyloid and all this sort of stuff with drugs and all that. Didn't fix the problem, did it? Nah. So basically now they've come up with this new idea. Let's create transgenic mouse. And a lot of the stuff, the recent research and recent drugs that have been coming out don't really do much at all. The problem is getting worse because it's a metabolic disease. People are eating too much shit. Simple as that. But, people want, these companies want to say, well, you can eat your shit and we'll drug you, So standard MD model hasn't changed. We know that. And so that's where this sort of bullshit research comes from and will be continuing to come from this sort of bullshit approach. So please do not fall for these stupid types of, studies that try and actually BS you. A transgenic mouse is not a human. There are fundamental differences. One gene does not make it human. And the results are not going to be similar to humans. If 40% similarity cannot tell us anything, how can just because you make it 100% of that gene, but all the other genes are 100% different, how's that going to be similar? Not going to be. So, yeah, guys, I mean, fuck me. You got to realize that the research is quite dodgy. Obviously, we can't experiment on humans. Very difficult. But we could do certain types of studies with nutrition to see in regards to these sort of ailments. But that would mean it would be exposing people to real food. Shit. Which also means that it would actually show that it actually can produce better results than even their fucking shit expensive drugs. We can't have that. Now, can we? Anyway, back to the normal noise, background noise. Anyway, hope you enjoyed it. See you soon.